Hello, people on Zoom. There's more of you on Zoom than in person, so thank you for coming. So my name is Joselynn. I'm a genomics data scientist at the Computational Biology Core and the Center for Computation and Visualization. And today I'm going to talk a little bit about how you can use R and Seurat to analyze your single-cell RNA-seq data. Let's see. So I've got everything in GitHub at this repository name, which is quite long. I'll paste it in the chat. That might be easiest, actually. Sorry, I keep losing my mouse. Chat. There it is. So that's the GitHub repo. So basically, the first thing that you're going to do was, oh yeah, we're doing all this workshop on Oscar. So if you don't already have an Oscar account, you won't be able to follow along. But if you clone this repo and you go into notebooks and _book, you can open these HTML files and just kind of follow along that way. If you do have an Oscar account, you can go to ood.ccv.brown.edu, which is this page here. And then you're going to go to, on the top of the blue menu bar, you'll see a thing that says clusters. So click on that and then click on the dropdown that says Oscar Shell Access. And then you should already be in your home folder as soon as you open this, but if you're not, go to your home folder and then clone this repo by pasting this command. We're also going to use Singularity for this workshop. So you can, if you look under interactive apps in that blue menu bar, you'll see you can click on this one that says RStudio Singularity, which is RStudio Singularity right there. And then I've put in this repo all the things you want to put in these fields. So basically, most of these things are going to, our account and partition are going to be blank. Number of hours, we can just say 2. 1 core will be enough. 100 gigs of memory should be plenty. And then I've also pasted this path to the Singularity container in this repo. But basically, all the packages we need will be in this container. So just copy and paste that full path there. And then you shouldn't need to put anything here. Just leave that empty. And then hit launch. It starts, you can click this button that says connect to RStudio server. Fine. And then you're going to go over here to File, Open File, and then go to the folder where you cloned this repo and go into the, you're going to go into the Notebooks folder and then open up the one that's called intro.qmd. Yeah. You can even, from the very beginning, you could just go up here and click Run All, which is what I'm going to do right now. So then it'll start running this code while I go through some talky bits at the beginning. Okay, so go to, maybe this mode will look less ugly. Let's see. Okay, so we already went over this about how to use the notebook. So we're going to talk about Seurat objects and what those look like and how you can interact with them. We're going to talk about data QC and filtering. We're going to talk about using SC-transform to normalize your data. And then we're going to talk about clustering and dimension reduction. We'll talk about data integration, differential expression, and data visualization. Do you guys have any questions? You good?
Our R student didn't start yet. We wait for a minute.
Oh, your job didn't start yet. You're still on this
Yeah.
menu. Okay. You can just follow, if you already cloned the repo, you can follow along with on like the HTML file. Are you able to open that?
Didn't.
Your job didn't start?
No, it didn't. Okay. Yes.
Yeah, and then go to the.
How about in the chat? Are people having technical problems in the chat? Are you good to go?
Yeah, it's not.
Oh, I see something in the chat. Let's see.
Yeah.
Yeah.
Having issues as well. Oh dear.
Go back to, no, just scroll down.
See if I can move this over here.
I'm good.
Why don't we just do something else then? Let's do,
Yes.
if you are having trouble, which it seems like many of you are, I would recommend that you just open up a terminal on your laptop, go to like your home folder, and then just clone the repo. Oh, it already exists, so that's fine. And then if you open this folder,
Oh, perfect. Yeah.
you can go to notebooks _book,
I did get on you. Lose it.
and you should just be able to open this and like follow along this way. You won't be able to run the code, but you can at least like see what the outputs would look like. What was the issue, Eric?
I'm not actually sure, the R stuff.
It just started.
Singularity session just crashed.
Oh, okay.
When did you open that?
Yeah.
What was
The intro.umd. So go to wherever you.
Yeah.
Yeah. Intro.
Oh.
No faults. And then intro. And then you just say.
Great. Okay. So the first thing we're going to talk about is like a quick overview of Seurat objects. The first thing I want to mention, this is going to become less and less relevant as we go, but in November of 2023, Seurat made a pretty big update to Seurat version 5. So if you have like legacy code from some grad student who graduated in 2022 or something, and you're trying to run their code, and they're using the older version of Seurat, and you have a new version of Seurat, you're not going to be able to reproduce their results unless you use the older version of Seurat. If you come to our office hours, we can help set you up with a reproducible version of whatever version of Seurat you want to use. But just a caveat that there was a pretty big update there. And like I said, we're going to focus on using Seurat to analyze your data. And so I'll try to sort of highlight the differences between those two versions as we go, but it's not like an exhaustive description of the differences. The first thing we're going to do is set the lib paths, which you won't usually have to do, but this is just basically telling R that it should look for packages only inside the singularity container. And then I'm also going to set a seed at the start of the notebook so that we can reproduce our results if we decide to rerun this entire notebook. And I'm also setting this future globals max size variable. If you look at the Seurat vignette that I linked to above, they'll explain why we do this. let's see. Yeah, most of this notebook is taken from the Seurat vignettes, the GitHub repo, and this other like wiki that had a nice description of the different integration methods. Okay, so we set the seed and this future globals thing. Then you can go ahead and import all of your libraries. Two of the packages we're loading are these Seurat data packages, which basically are just like pre-populated sort of toy data to sort of start experimenting with. And here's a sort of a schematic of what a Seurat object looks like. You've got all these different slots. I think it's a little bit smaller so you can see the figures a little more. So you've got assays, metadata, graphs, reductions, tools, miscellaneous, and version. I feel like the ones I use the most often are assays and metadata. Assays is basically just a list of all the assays in the object. When you first make a Seurat object, it's only going to have RNA. Once you do the SCT normalization, you'll have another assay that's called SCT. If you have like multimodal data or something, you might have another library that's like antibody-derived tags or something like that. And you can see a list of all the assays using this assays function. You can see what assay is the active assay by looking in the active assay slot or layer in Seurat v5. And you can switch between them using this default assay function, which we'll do that in this notebook. Each assay will store different transformations of the data in different slots or layers, depending on which version of Seurat you're using. In the case of the RNA assay, these slots are the counts, which is just the raw counts, the data, which is the normalized counts, and scale data is like the scale data for dimension reduction. Let's see. And for all of these assays, they're all stored as a sparse matrix where the rows are genes and the columns are cells. In Seurat v4, you could access the counts using this get assay data function, which will still work in Seurat v5, but you'll get a weird warning message.
Yeah, it's
So you
so
can use
cold
the
in
layer
here.
data function or use
I
this,
mean,
like-
I need to be smart like that and bring a.
Oh, what was that?
I'm so sorry.
It's okay. Did you have a question?
No, I'm sorry. We were just saying that it was cold in our lab. I thought I was muted.
You're all good. Yeah, you can stop me anytime to ask a question. Okay. Yeah, so you can also access the sort of counts using this like double square bracket notation here. That'll work in either version of Seurat. Metadata is pretty important because this is basically a giant data frame of all of the like cell level metadata. So it'll include information about like what conditions, like if you have control versus treatment, metadata will tell you for each cell whether it came from your control versus treatment condition. It also has all this stuff that's going to be relevant for your QC, like N count and N feature RNA, which are N count is a total number of molecules or UMI detected, and N feature is a total number of genes detected. Once you've done clustering, you'll also have information about like which Seurat cluster or whatever each cell belongs to. And then you can use this like, you can just do like use table with idents in your Seurat object to get like a quick summary of how many cells are in each of your groups. And then graphs is just a list of all the nearest neighbors. There are cell by cell matrices containing the neighborhood overlap between each cell and its nearest neighbor. Reductions is just like a list of these dimension reduction objects. So it'll be like PCA, UMAP, etc. And then version will tell you which version of Seurat you're using. And then there's this other like tools and miscellaneous slots, which tools will just be populated by like, if you use some specific tool, that'll go in there. And then you can put your own whatever information you want in that miscellaneous slot there. Okay. Like I said, we are using Seurat data. So these like pre-built data sets. You can run this function to see which packages we've installed. Let's see if this works. Ooh. There we go. For some reason, it's not running in the notebook, but you can see it down here that we've got this IFNB data set and the PBMC3K data set installed. If you Oh yeah, it's running it down here. So you can see if you run like these little question mark, if you run this question mark code chunk, it'll show you this help, which give you some very detailed information about the data. So IFNB dataset is 14,000 interferon beta stimulated and control PBMCs or peripheral blood mononuclear cells. And then the PBMC3K data is just more PBMCs, but it's only 2,700 of them. You can also see that it stores stuff in layers. Again, they can store the raw or normalized data. And then you can also check out what's going on with the metadata here if you run this chunk. I don't know why this is not. So you can see that you've got like the original ident, so where if it's control or stim, you can see the end count and feature RNA, and then this one came pre-annotated with some Seurat annotations. If you run the same thing on the PBMC3K data, you'll see that you've only got PBMC3K data. There's no like control versus treatment in that one. So it's more likely that you will not be using a pre-made Seurat data package. You'll probably have to import your own data into Seurat. So I don't actually run these functions because we're not working from raw data, but you'd use either read 10x or read 10x H5 to import your data. 10x H5 works with these hierarchical data format files, which if you run like a multimodal 10x run, your data will probably be in this format. If you're just doing like the bog standard gene expression run, then you will have a folder that has, after you run Cell Ranger, you'll have a folder that's like your matrix, your features, and your barcode TSV files. And that is what you give to read 10x. You just give it that whole folder. And then after you've used either of these functions to read in your 10x data, then you use the create Seurat object to make your Seurat object. It's pretty straightforward, and the Seurat vignette explains how to do this. As always, if you have questions, you can come talk to us. Okay. The first thing we're going to do is subset these data sets so that they're smaller, so they only have 500 cells each, because otherwise this workshop would take too long. And then the next thing we do is merge these objects. Basically, we just do this to make it easier to do all our QC. It's not like a super sophisticated way to like integrate your data. It basically just like slaps the matrices together. It's not, you don't actually want to work with merge data other than to do the QC. Yeah, and you use this merge function, and then you can add cell IDs to basically make sure that the sort of project ID is stored. So it's like prepended to the beginning of each cell name. And the original project ID from each object will be stored in this original ident column of metadata. First thing we're going to do is look at the percentage of reads that map to mitochondrial genes in each cell. The reason we do this is because basically cells that have a lot of mitochondrial reads are usually like dead or dying cells, so we want to filter them out. The first thing you want to do is you can run this like grep command to figure out how the mitochondrial genes are named, because sometimes the MT is capitalized, sometimes it's not. Sometimes the M is capitalized, not the T. So run this grep command to try to sort of figure out what format your mitochondrial genes are in. And then I don't know why these are like, some of these code chunks are getting like collapsed, but basically this little next chunk is adding the percent mitochondrial reads to the metadata, which you'll be able to see if you go to source code. I bet you'll be able to see the code chunk. I think just running it in visual mode looks nicer, but some of the chunks are getting lost in the shuffle. The next thing we're going to do is we're going to use that idents function that I talked about before to specify that we want to use the original ident as like our main identity class that we care about. And then we can manually set the levels to tell it exactly what order we want things plotted in. And then you can make some of these violin plots that basically will, go back to this mode. I don't like the fact that it's like hiding all of these things. Okay, here we are. So basically on the x-axis will be your different identities from your idents, and then on the y-axis is your n features, which is just the number of genes. Same thing with the n count, which is the number of UMIs, percent mitochondrial reads. And you can sort of see in this figure that these IFNB samples look a little funny. And this sort of confirms it that I think they did some pre-filtering before they made this object, so there's no mitochondrial reads in these. You can also use this feature scatter function to sort of look at the relationship between these different QC metrics. And again, you can see that the percent MT is basically 0 for both of these immune samples. I really like to actually just use ggplot to sort of visualize all these things at once. So you can make your own little data frame of all the QC metrics that you care about and sort them so that by the percent mitochondrial reads, and then just use ggplot to say, put the n count on the x-axis and the y-axis is the n features, and you can color the dots based on the percent mitochondrial reads. And this is nice because you can see that sort of the reads that have high mitochondrial content usually have the lowest n feature and n count. And the things you want to look for in these QC plots are the following things. Low quality cells or empty droplets might have very few genes. Dead and dying cells will have high mitochondrial counts. Doublets or multiplets might have really high gene counts. The total number of molecules in a cell should generally correspond with the number of genes. And that's true, right? And we can also make some observations about our data specifically. Most of the cells have less than 2,000 genes and less than 7,000 or so UMI. Again, it looks almost like the immune stim versus control data has been pre-filtered. And so, your goal in filtering this data should be to sort of retain as much useful information as you can while trying to sort of remove some of these doublets, dropouts, or dead cells. And so we're going to pick some thresholds for our filtering based off of what these plots look like. But if you're working with your data, it will definitely not look like this. So you shouldn't just copy and paste these thresholds that I've picked. They're not like a hard and fast cutoff. The other thing I want to mention is that I don't really dig into ambient RNA at all in this workshop because it's usually something that you deal with before the Seurat stage. But that's another QC consideration that you might want to think about. And I pasted a link to 10x has a nice little document on sort of why it happens and what some tools that you can use are to fix it, and also how you might detect it in some of the outputs from Cell Ranger. Okay, so we're going to use subset to filter our data. And we're just going to keep cells that have between 500 and 7,000 N features and less than 5% mitochondrial reads. And then if you want to, you can kind of re-examine your QC plots. And you can look to see how many cells you've actually lost with this filtering. We lost about 50 of the PBMC3Ks. Okay. Next thing I want to talk about is normalization. So normally, if you just run the standard Seurat pipeline, they usually have you do this series of steps called normalize data, find variable features, and scale data. We almost never use this pipeline. We basically always use SC transform. I'll talk a little bit about when you might not want to use SC transform. But basically, it's pretty similar to EdgeR and DSCoV that people use for bulk RNA-seq. It also, it will pool information across genes to help you get, to help sort of address the sparsity issue from single cell data. This is not rendering. Oh, there we go. So this figure in particular is a really nice one. It's right from the SE Transform paper, and I think it illustrates why we like it so much. So in the panel on the right, the B panel, you can see on the x-axis it says % of variance explained by sequencing depth, and on the y-axis there's just a few different cell types. And the different colored bars represent log normalization, which is like the standard Seurat normalization method. And then the Pearson residuals bar is the SE Transform normalized data, and you can see that basically SE Transform does a better job of controlling for variance that is explained just by sequencing depth alone. I will say that you should not use SE Transform, or you should proceed with caution. If you have a situation where your data is very heterogeneous and you expect that a high proportion of your cells will have significant biological variation across your samples, right? So like maybe you're working with some, this seems so unlikely, but maybe you're working with like some cell atlas where it's like every single tissue from a mouse, and that your data set is that you have like, you've sequenced 10 tissues from a mouse, and that's what you're trying to normalize all together. Then I might, I would try both and see kind of how things look, but I would not use that. SE Transform doesn't seem like the best tool for that. Again, this is like an issue between Seurat v4 and v5, but different versions of Seurat run different versions of SE Transform. That's another thing to like watch out for if you're using someone's like legacy code. Some of the results you get will be different because they tweak some of the ways that SE Transform works in the different versions. So, be careful. The other thing you might see when you run SE Transform is this warning that says iteration limit reached. I've actually only ever seen this with the older version of Seurat, so maybe it doesn't happen now. But basically, if you get this warning, you could ignore it because the parameter estimation that's generating that warning message gets regularized later anyway. You can use the vars to regress argument to sort of regress out any nuisance variables like cell cycle or batch effects or the % mitochondrial reads. By default, SE Transform will only return data for variable genes in the scale data slot. You can turn that off by saying return only var genes equals false, this argument. Yeah, I do this often. I do this often because sometimes people will come in to the analysis with some theory about or some gene of interest that they want to look at. And when I run SE Transform, we lose it because it's not actually highly variable in their data. So if you have some gene of interest and you run SE Transform using the standard, default parameters and your gene is missing, this might be why. I turned it off in this particular workshop because it runs much faster if you leave it on. Oh yeah, and the first thing that you have to do actually, so in previous versions of Seurat, they would make you turn your merged Seurat object into a list of Seurat objects. Newer versions of Seurat don't make you do that, but they do make you split, which is what we're doing here. We're splitting based on the original ident. And then you can run SE Transform, which is what we're doing here. And then you can run the PCA. Uh-oh. Oh, I think this is because I actually already ran this. And then you can make an elbow plot. Nope. Let's run all these things above. I already did it, so it should be fine, but okay. You can't actually see the plot because I'm rerunning the code, but basically once you make this plot, you'll see that there's diminishing returns once you get above 10 or 15 PCs. Like your elbow plot will sort of start to flatten out a little bit, and that's how you know you've reached the point where more PCs are not giving you more info. And you can use that when you do your clustering later. Okay, so it's running SE transform right now. I think. See if this works now.
Oh,
There we go. So now the PCA is running. And then you can see that your elbow plot is starting to really flatten out at around 10 or 15 PCs. Okay. The next thing I want to talk about is integration. You almost always are going to want to integrate basically across your different batches or donors or sequencing runs or conditions. And the reason you want to do this is because it helps you to sort of match up like the shared cell types or cell states across your different samples. And I'll show you what the unintegrated data looks like later, and maybe that will convince you that you should do this every time. Seurat v5 uses this integrate layers function to sort of let you play with these different methods for running the integration. And part of the reason they do this is that some of them are like more conservative than others. So depending on depending on your data set, some of these might work better than others. So the 4 methods that Seurat that ships with without you having to install anything extra are the CCA based integration, which is canonical correlation analysis, reciprocal PCA integration, joint PCA integration, or harmony. The first 3 are like anchor based methods, which just sort of relies on finding pairs of cells or anchors that are mutual nearest neighbors. Harmony is a non-anchor based method because it does soft clustering and iterative correction. Okay. CCA is sort of the one that was like the default integration in Seurat for a while. Basically, it will perform canonical correlation analysis between pairs of data sets, L2 normalize the cell embeddings, identify mutual nearest neighbors between datasets in the CCA space, and then use those anchors to correct the data and align the matching cell populations. CCA is well suited for situations where all the samples that you're integrating have like the same cell types in every sample. If you have a situation where like you know for sure that one of your samples is missing one cell type, or it's like cell sorted and you only have endothelial cells or something, I wouldn't use CCA because it's going to like overcorrect your data. If you don't have that going on, it's probably totally fine. If you do have that situation, I have used reciprocal PCA for this, and that worked great. So basically, it will compute PCAs on each sample separately, project each data set into each other's PCA space, normalize the cell embeddings, identify the mutual nearest neighbors, and then use those anchors to correct the data and align the matching cell populations. I recommend this in situations like I just described, where you have like some cells, some significant fraction of cells in one data set that doesn't have another matching type in another data set. Or if there's like if you have a really big data set, I think it works well for that. Yeah, and if they are from the same sequencing platform, this is probably fine, because this is sort of CCA will definitely, you're running into a situation where you might be like over integrating and you're like disguising your biological signal, which is less of a concern in our PCA. I've actually never run joint PCA because I've never had data that sort of like meets the criteria of when it could work well. Basically, it combines all the data sets into one big matrix, performs PCA in the combined data, normalizes the cell embeddings, finds neighbors in the joint PCA space, and then uses those anchors to correct and align matching cell types. It's often faster than CCA or RPCA, but you really only want to use it in situations where like your data is already like somewhat aligned or won't have any minor batch effects, which again, I've never actually seen in practice. Harmony is the last one we're going to talk about, and basically it performs PCA on the combined data sets, applies soft clustering to group the cells by similarity, iteratively corrects those cluster assignments and cell embeddings to align the data sets, and uses a ridge regression to minimize the effect of data-specific variation. It is generally faster than any of the anchor-based methods, which we'll see when we run these next few code chunks. and it performs well on situations where there's like complex batch effects. I've used it in situations where like multimodal data before 10x made the kits where you could do like ATAC-seq and RNA-seq on the same cells. I had two separate libraries that were done separately of like single cell RNA-seq and single cell ATAC-seq. I used Harmony to integrate them and it worked great. So that's my plug. Okay, so then we'll run all these methods. First, we'll run CCA. Note that we're including this normalization method equals SCT argument because we didn't use the bog standard Seurat normalized data, scale data, find features, whatever. I literally have never run that method. So, okay. CCA ran. Now we'll run the joint PCA. We'll just queue up these next ones.
Jocelyn,
Yes.
is there a time that you think of where you want to use CCA or RPCA over Harmony based on how well Harmony seems to work?
Eric asks if there's ever a situation where you'd want to use CCA or RPCA over Harmony. I'm not sure. I mean, my advice is always to try all of them and see kind of how things shake out. I've actually only ever used Harmony in that situation where I had like the multimodal data sets to integrate, but I should see how it compares to RPCA with some of these like data sets where I had like there was one data set where we were basically comparing like cell sorted to non-sorted, and Harmony might have been a good choice for that. I should try and see. Yes, kind sir.
When you say try all of them and
Yeah.
see,
Yes.
is that when doing that, do you have to do something like equivalent of like type 1 error correction? Something like that?
Oh, Paul, stay. I don't know. Paul asks if we need to do some sort of type 1 error correction if we're playing with all these different integration methods, and I don't know the answer to this question. I feel like I should ask August and Jordan about this because they'll know. I don't know. I basically I try all these different methods. I do a lot of visualization and exploratory data analysis and, try to pick the one that seems the most consistent with the expectations of the people who know about the system. If I bring some of my, like in this data set, for example, where I had the cell sorted in the pooled data, when I bought back the CCA integration to Lalit, he was like, this looks totally wrong. Something is not right. So he knew when looking at the clusterings that something funny was happening. So I said, okay, let's go back to the drawing board. And that was why we tried the RPCA. So I think like it isn't a super like statistically rigorous answer, but usually it's just, in my case, it's like consult with someone who knows the system and see if it seems reasonable and go from there. I go into some other detail a little bit later about how to like how you could QC your clustering. So maybe like looking at the clustery clusters could help figure out which integration method is best. I don't know. These are all excellent questions. I do want to draw your attention to the fact that I think CCA. So CCA ran in 10 seconds. Joint PCA also ran in 10 seconds. Harmony ran in 1 second. And RPCA ran in also about 10 seconds. So Harmony is much faster. Okay. Clustering. Seurat will group your cells into clusters. Basically, if the cells have similar expression patterns, I'll cluster them together. The first step is find neighbors, which will construct a k nearest neighbors graph based on the Euclidean distance in PCA space, or find the edge weights between any cells based on the shared overlap in their local neighborhoods. And then to the cluster the cells, we'll use find clusters, which will apply the Louvain algorithm to iteratively group the cells together with the goal of optimizing the standard modularity function. Find clusters takes a resolution argument, which we'll talk a little bit more about in a little bit, but basically it defaults to a value of 0.8. If you want more clusters, use a value above 1. If you want less clusters, use a value below 1, but generally setting it between 0.4 and 1.2 is usually okay. As long as you don't have more than like 3,000 cells. If you have a lot of cells, then you might need to sort of adjust these expectations. So we can run all these, you can run this stuff on all of these different, like we can run on just the PCA space and get some unintegrated clusters. We can run it on the CCA reduction to get some CCA clusters, et cetera, et cetera. So we'll run the clustering on all these different integration methods. Just gonna close this. And then you can make some UMaps, which is basically just a dimension reduction technique. And then thinking. This is too long. And then basically we're just going to make a bunch of dim plots where we plot these different reductions. And then basically I'm just using patchwork to kind of combine all of the plots together. Put it right here, make this a little bit bigger so you can actually see. This one right here is the unintegrated data, and you can sort of see that like the colors of the dots are these like the original idents. You can see that if they're unintegrated, you have these like clusters of cells where you don't have good integration of your idents. So it's going to be really hard to run differential expression on this cluster because you don't have all the samples sort of represented. And you can contrast that with basically any of these other integration methods look good. This is sort of a bad example because these are all PBMCs. Your data probably won't look this nice in real life. But I think it at least shows you why you should integrate. Okay. The last thing you want to do is rejoin the layers using this join layers function. You need to do this before you can run differential expression, but you can always resplit the layers if you want to rerun any of this stuff. You can also so these data sets came pre-annotated. You can also make one of these little tables where you see how well the clustering and your cell types agreed. Just keep in mind that we were pretty aggressive about our subsetting and we only have 500 cells in each data set. So things don't look as nice as they could, but basically the columns are the RPCA clusters and the rows are those pre-annotated cell types. I don't go into how you can annotate the cell types in this workshop, but I talk about it when we do the research computing bootcamp. I give a more in-depth version of this workshop where I talk about how you can use some of these machine learning methods to kind of automatically annotate your cell types. And this is if I'm doing that, this is kind of one of the first QC things I do is look to see how well the clusters and the cell type annotations agree. You can also just make some dim plots where you look at the sort of labeled cell types. This is sort of a mess because there's a lot of cell types. And you can compare that to the clusters labeled with the Seurat clusters. So you can see that in some cases, Seurat has all of these B cells, B activated, and PDC cells all in one cluster. I don't actually know what PDC cells or B cells are, so I don't know if that makes sense, but I assume they're similar, and that's why they're clustered together. Okay. The other thing I really wanted to talk about really quickly is clustry, which is basically a nice package that you can use to get a sense of how tweaking that resolution parameter will affect your clustering. So basically, you're going to set a range of resolutions and then run the clustering across a range of those resolutions. And then clustry will make this nice little clustering tree for you. Where you can see at the top, when we have resolution is 0, we just have one big cluster. And then as you increase the resolution, you have things splitting out to more and more clusters. If you have a situation where these plots start to look real messy, where stuff like splits out and then reclusters again, and things start to look really hairy, that is like a sign that you might be getting into like over-clustering territory. So it's like a nice way to sort of see what clustering thresholds seem reasonable. Okay. I'm going to try to talk really quickly about differential expression analysis. We don't have a ton of time left. Basically, most of Seurat's differential expression features can be accessed through either find markers or find all markers functions. By default, Seurat will use the Wilcoxon rank sum test to test for DE genes between two specific groups. You'd use find markers and just specify ident 1 and 2, which are the two groups you want to compare. And then you can use find all markers to basically compare each ident to every other. So like my usual use case for this is like I use find all markers to find cluster markers. Say like, okay, show me the markers for cluster 0 compared to clusters 1 through 9 or whatever. One thing I will say is that if you have like a super complicated experimental design, Seurat might not be the best choice because it doesn't have, like for example, I don't think you can run Seurat with like an interaction term. So if you have like a more complicated experimental design and you need help running your stats, come talk to us office hours. We can help you sort that out. Okay. Since we normalize using SC transform, we have to run this prep SCT find marker step first, which basically just like recorrects the UMIs for us. Do that here. We're switching. We're specifying that we want to test for differences between the original ident groups, not the Seurat clusters. Then we run prep SCT find markers. We specify that we want to use the RNA assay for this test. And then we run find markers to compare the stim versus control. And then the output is like a p-value, a log fold change. PCT1 and PC2 are like the percent of cells where the feature is detected in the different groups. So like PCT1 is stim and PCT2 is control in this case. And then there's like an adjusted p-value column, which is based on the Bonferroni correction using all features in the data set. The log2 fold change column will be positive if the feature is more highly expressed in the first group. So more highly expressed in immune stim in this case. If you omit ident 2 when you run find markers, it will sort of automatically do this find all markers-esque thing where it will automatically just test for differences between your first ident and all other groups. Another useful one is you can set this argument only pos equals true to basically only find genes that are like highly expressed in your ident group 1. And this is just showing an example of that here. And then I kind of alluded to this before. We can also look at the cluster markers. So we'll use this idents function to switch to say that our primary ident is the RPCA clusters. And then we can run find all markers to find the markers distinguishing the different clusters. And then you can use some dplyr wizardry to look at like the top 2 genes per group or something by based on the average log2 fold change. Oh, let's see. You can make these feature plots to look at expression of specific genes, which I think is kind of helpful. Here I'm saying that we want to use the SET assay. The feature plot will by default plot the data slot from the default assay. So we're applying the data slot from SC transform here, which is, I believe, oh, yes, it's the log1 p counts. I love that for me. You can also make these heat maps in Seurat. I don't love the Seurat heat maps, but I'll show them to you anyway. You can also you can customize the Seurat figures just like you would customize a ggplot figure. So you can use custom colors or whatever. I don't want to dig into this too much because I think there's going to be some R workshops later on in the semester. Oh yeah, you can change sort of what legends are shown, because this having both the dots and the color bar feels a little bit redundant. So you can just say only show me the color bar at the top, which this is just the different Seurat clusters. Another one that I really like is the dot plot. I actually think I like this more than the heat map, where basically the size of the dot indicates the percentage of the cells expressing the feature, and the color is the average expression level, and it uses scale data by default. By default, it uses this, in my opinion, kind of weird gray and purple color scheme. You can customize it on your own if you feel so inclined. That's all I have. Now you know about how to use Seurat. If you have questions, you can always stop by our Zoom office hours. You can look at the CCV calendar if you want more details. And then you also can scan this QR code or click the link to do, basically there's a survey so we can keep track of who all is coming to the DSCoV. So if you could fill out this survey, that would be just lovely. Any questions, thoughts, concerns, comments? Great. Stop share.